Good morning or afternoon, everyone, and welcome to today's webinar. My name is Marie Latulip, and I am pleased to serve as the Director of Science Programs at the Institute for the Advancement of Food and Nutrition Sciences, or IFANS. And before we start, just a quick note on the organization that's bringing you this webinar today. We are a nonprofit organization focused on advancing food and nutrition science for public benefit. And the way we do this is we connect our tripartite membership, so that's scientists from government, industry, and academia, to support collaboration and to catalyze knowledge. And what we do is we fund what we like to call actionable research, and that's typically conducted by external researchers who are selected through a competitive process. And you can actually find information about all of our funded projects on our website. We advance food and nutrition science for public benefit by convening experts across these sectors to engage in dialogue. And the way we do our work is that we leverage pooled industry funding, and then we combine that with in-kind contributions from both the public and the private sectors. This slide shows the overall mission and focus areas of our Gut Microbiome Committee, and that is the committee that brings you this webinar. Today is the second in our series focused on the role of diet and gut microbiome in immune response and inflammation. And our intention here is to explore these relationships from different perspectives through the series, understand the state of the science, as well as gaps that could be addressed through IFANS programs. So hopefully you see a control panel on your screen like this one. And we encourage everyone to just type your questions as the presentation goes along, and we'll take those at the end. And your questions are only visible to the IFANS staff, so essentially they're anonymous when we bring them forward at the end. So with that, I am quite pleased to welcome our speaker for today, Professor Philip Calder, with whom I'm sure many of you are familiar. Dr. Calder is the head of School, Human Development and Health, and Professor of Nutritional Immunology within Medicine at the University of Southampton in the UK. And he has quite broad research interests in nutritional modulation of immunity, inflammation, and cardiometabolic disease risk. So much of his work has been devoted to exploring the metabolism and functionality of fatty acids with an emphasis on the roles of omega-3 fatty acids. So you'll see some focus on omega-3s today. He has over 700 publications. He's listed by Claravit as a highly cited researcher, and he's been the recipient of many awards and has served on numerous prestigious committees, as well as editors and associate editor of many top-tier journals. And he's currently associated for 2 key nutrition journals. So Professor Calder will speak. We'll open it up to public comments and questions. And then at the end, we're going to have a closed session with the Gut Microbiome Committee so they can really explore possibilities for work within that group. So that'll be the logistics. So with that, we turn the platform over to you, Dr. Calder.
Great. Thanks very much, Marie, for inviting me to speak today, and thanks very much for to everyone who's shown up to listen this morning, this afternoon, this evening, depending on where you are. So I'm going to try to address this question about whether diet and dietary components can impact gut microbiota and how this relates to supporting a healthy immune response. First of all, these are my disclosures. So I'm going to give you a brief overview of the immune system, say something about the role of the immune system in health and disease, something about the gut microbiota link with immunity and inflammation, then talk about probiotics, what I've called classic prebiotics. These are usually short-chain oligosaccharides, what I've called emerging prebiotics, then say something about foods and diets towards the end, and then summarize, conclude, and throw out some discussion points. I've got a lot of slides. I'll try not to go too fast, but I also want to get to the end of my talk in about 30 minutes or so. So the immune system is a cell and tissue system that protects us from harmful organisms that invade the body. We call these harmful organisms pathogens, things like harmful bacteria, viruses, and so on. A well-functioning immune system is key to providing good defense against pathogens, and we know that because people who are immunocompromised or immunosuppressed, so with weakened immune systems, are at much greater risk of being infected and of infections becoming more severe, perhaps even fatal. So there is a link between robust immunity and protection against infection. Here I show the 4 general functional features of the immune system. So first of all, it acts as an exclusion barrier to keep pathogens out. Secondly, the immune system can recognize pathogens if they breach the exclusion barrier and can identify them as being harmful or harmless. Thirdly, the immune system acts to eliminate, that is, to kill pathogens that it's identified as being harmful. And then finally, there's a component of immunological memory so that immunological encounters are recognized for quite a prolonged period, maybe even for the whole life of an individual, once the initial immune response has happened. So that gives us better protection if we get reinfected. And of course, the memory response is the basis of vaccination, which we've all become very familiar with over the last couple of years. So the immune system is very complicated, it's very sophisticated, it can recognize, it can eliminate, it has memory. This sophistication comes about because of the many different, mainly cellular components of the immune response. These are generally divided into innate immunity, that includes the barrier functions and the inflammatory response, but also some cellular responses, for example, phagocytosis, which is the engulfing of bacteria by phagocytic cells. So that's innate immunity. And then we have acquired immunity, which involves T lymphocytes or T cells and B lymphocytes or B cells. Innate immunity is not very specific, but it can be very rapid. Acquired immunity is highly specific to a particular individual antigen, but takes a period of time to develop. There's links between innate and acquired immunity. One of the key links is that antigens that are derived from the digestion of phagocytosed pathogens are presented to antigen-specific T cells, and that activates the acquired immune response. So antigen presentation is a link between innate immunity and acquired immunity. Conversely, T cells in particular produce many different cytokines. Cytokines are like hormones that act within the immune system. These cytokines influence other T cells, they influence B cells, but they also influence the activity of phagocytes, natural killer cells, and so on. So this is regulation of innate immunity by cytokines produced as part of the acquired immune system. Just worthwhile mentioning in passing that B cells are the cells that produce antibodies. So different types of cells have their individual functions, but they all have to work together to provide robust defense. So I sort of capture this in this simple cartoon about antiviral defenses, just showing all the different immune cell types that are working together to make sure we're protected against viruses. So immune cells have their own functional roles, but they have to work together in a coordinated, integrated way to give us good host defense. But again, just to go back to basics, we have barrier function to keep pathogens out, and then we have the cellular components of innate and acquired immunity if pathogens breach the barrier. And this is where the identification, elimination, and memory become important. As I mentioned already, weak immunity means we have poor defense against harmful organisms. That results in us becoming infected and infections becoming more severe. And of course, in the last, or certainly in 2020, weak immune systems were exposed as a major public health challenge. That was a bit of a surprise to a lot of people. But of course, the importance of weakened immunity in susceptibility to infections has been known for decades now and is already a major public health problem in some geographies. This on the left is actually a poster that you could download for free and display, and I really like it because it captures what I've said so far. So you could stick this on your window or your door. Someone in this house has a weak immune system. They're more likely to catch coronavirus and more likely to suffer serious complications. So this is true, and it's exactly what I've just been saying, but put into, if you like, a layman's perspective. One bit of the immune response that I mentioned in passing is inflammation. So inflammation is triggered by exposure to microbial components, so it's triggered from infection, but it also can be triggered from tissue damage, metabolic stress, oxidative stress. People have become very interested in inflammation because although it's designed to be helpful, it actually can also be harmful. The 5 cardinal signs of inflammation are heat, redness, swelling, pain, and loss of function. So inflammation is a physiological response. It's part of innate immunity, but we know when inflammation is uncontrolled or excessive, if it continues without its normal resolution, we can get tissue damage, pathology, metabolic changes, and these can be damaging. So there are lots of diseases linked to inflammation. Some of these are linked to what I call high-grade inflammation, and you would treat those diseases often with anti-inflammatory drugs, things like arthritis, inflammatory bowel diseases, asthma, and so on. But there are also many common diseases that are linked to low-grade inflammation and the inflammation that occurs as we become older. Things like vascular disease, metabolic disease, cognitive decline has a low-grade inflammatory component, some cancers, changes in body composition, and even immune decline actually is driven by low-grade inflammation. So if you put all this together, it seems the immune system has 2 faces. One is this protective face, where we have defense against harmful microbes, giving us resistance to infection. And the other is this harmful face, defined by excessive or ongoing inflammation, causing tissue damage, pathology, disease, and in some cases, even death. So the system has got to balance up here. We need protection and we don't want harm. And I think physiologically, in a healthy person, this is exactly where we'd want to be. We are resistant to infection and we don't develop the adverse aspects of inflammation. Now, one of the interesting things is, although we think about immune cells as circulating in the bloodstream, like the white blood cells, in fact, most of our immune cells are not in the bloodstream. Only about 2% of our immune cells are in the bloodstream at any one time, and about 70% of our immune cells are linked to the gut wall. And the reasons for this are probably pretty obvious. One is, of course, the gut lumen is full of microorganisms. Some of them are pathogenic, so we have to be able to protect ourselves against pathogens in the gut. And the other, of course, is we have friendly bacteria, commensal organisms, and also what's left over from the digestion of food. And we need to be able to tolerate, that is, not mount an adverse immune reaction to those things that are not harmful. So the immune system in the gut wall is what gives us oral tolerance and what protects us against harmful components in the gut lumen. And the immune cells in the gut wall are organized into particular structures, like these Peyer's patches here. So the immune cells are communicating with one another, they're sampling the gut lumen, they're making immunological decisions. They actually move to other sites like lymph nodes that are nearby, and that's where they make even more immunological decisions and start responding either in an active or a passive way. Some of these cells actually will pass back to the gut wall and give us the immunity we need. Some of them go to other places, for example, to the lungs. So we have a direct link between the gut immune and the immune lung axis. So in fact, there's a gut-lung immune axis, if you like. The reason this is really important, of course, is the gut microbiota interacts not only with the gut wall, that is the monolayer of epithelial cells, but also with this gut-associated immune system. So there's a bidirectional interaction between the microbiota and the immune system, and between the immune system and the microbiota. And we think what this means is our gut microbiota plays a role in supporting and regulating our immune response, just as our immune system controls our gut microbiota. And part of this controlling of the immune response actually is controlling the inflammatory response as well. So I think there are bidirectional interactions all over the place here. So the microbiota is supporting and regulating the immune system, is regulating inflammation. I mentioned that inflammation is actually part of the immune system anyway, but in turn, the inflammatory response in the immune system are keeping the gut microbiota in check, trying to deal with any pathogens, trying to promote the growth of helpful commensals. So we can think of many different strategies to support immunity and to control inflammation. Some of these might be directed systemically, for example, through the diet, so that they're absorbed into the bloodstream and they act systemically once they're absorbed. And this in general is the way that vitamins, minerals, omega-3 fatty acids and so on are working. But another strategy, of course, is to target the gut microbiota and get the microbiota into a more healthy composition, and through that means support a healthy immune system, a healthy immune response, and help us control inflammation. And of course, the key things we would think of here would be fibers, prebiotics, and probiotics. So I'm going to talk now about a few studies of probiotics and then prebiotics. These are just examples of studies that are out there. This is a trial in healthy older people. It's not a controlled trial. Uses Bifidobacterium lactis, HN019, with a run-in period up to this point here, week 3, then an intervention over 3 weeks, then a washout period of 3 weeks. And what the researchers did here was they measured phagocytosis. This is this engulfing of bacteria by neutrophils, phagocytosis by monocytes, and natural killer cell activity. So during the period of probiotic intervention, these all increased significantly, although the increase actually was quite modest, but it was a statistically significant increase. And I think, although this is not a controlled study, there is an element of cause and effect here, because there was a 3-week washout period during which these immune markers returned back to baseline. Similar sort of study with the same organism, again in older people, this time with a control group, now looking at the production of a cytokine that's really important in antiviral defenses, interferon alpha. That didn't change with a 6-week intervention in the control group, but it increased over the 6 weeks with the probiotic. And then this is neutrophil phagocytosis, which again was shown to increase over the period of probiotic intervention, but no change in the control group. Now, I mentioned vaccination already once, and vaccination is really a challenge to the immune system. And you'll be aware, because probably most of you have had plenty of vaccinations in the last couple of years, that you have an immune response. That's actually one of the things that makes you feel ill after the vaccination. But you can measure the immune response in the laboratory by measuring antibodies that the immune system makes to the vaccine. So this is a trial that I was a little bit involved in, although the trial was done in Italy, of 2 probiotic organisms, Bifidobacterium BB12 and the Lactobacillus casei 431, compared with placebo. And these are the different types of antibody classes, IgG, IgG1, IgG3. Following vaccination in the control group in the light gray bars, and in the probiotic group in the darker bars, black in the top graph, darker gray in the bottom graph. And this is the comparison of BB12 with placebo, and this is the comparison of LKCI with placebo. And you see the antibody responses were higher post-vaccination with probiotics compared to post-vaccination with placebo. The vaccination here was a seasonal flu vaccination, by the way. This is a trial in older people, again using the seasonal flu vaccination, looking at another LKCI compared to control, delivered in a yogurt 100 people in each of the two groups, again using the seasonal flu vaccination. Now, there are three components to the seasonal flu vaccine, three different flu viruses, if you like, H1N1, H3N2, and B. So you can actually measure the specific antibodies to each of those three viral strains. So these are the blood antibody levels to each of the three viral strains at baseline and 3, 6, and 9 weeks post-vaccination in people, older people who receive yogurt with probiotic or yogurt without probiotic for 4 weeks prior to the vaccination. So you'll see if the individuals received the probiotic, the antibodies were always higher than in the control group, although this was only significantly so with respect to the B strain of the seasonal flu virus. So probably this study is underpowered as far as the H1N1 and H3N2 strains are concerned. Now you can measure something called seroconversion. So this is a readout of the change in antibodies to the vaccine over time. So the period of intervention here goes out to 60 days, and this is the seroconversion to the B strain. So in the control group, only 10% of people seroconverted, so they had a good response. This was doubled in the probiotic group, and the seroconversion remained higher in the probiotic group than the control group right up to 150 days. And then this is seroconversion to each of the three strains 5 months after vaccination. So the response to the vaccination was always better in the older people receiving probiotic compared to control. There have been several of these vaccine responses done. This is a meta-analysis of 7 trials of probiotics and the seasonal flu vaccine. Overall, there was no effect on titers to the B strain, despite what you just saw in that one study, but there was a significant increase in titers to the H1N1 and H3N2 strains. So the conclusion here would be that a probiotic intervention improves the immune response as measured by the antibody response to seasonal flu vaccination. So probiotics, at least some probiotic organisms, improve immune function. I've shown you some examples there. This would enhance defense against bacteria and viruses, resulting in fewer infections. Of course, because of their colonization feature, so they compete with pathogens, they drive pathogens out of the gut microbiota. Of course, if you have fewer pathogens, you're less likely to get gastrointestinal infections. So I think there's a double-pronged approach to probiotics and infection, but one of those routes is through improvement in immune function. Let's turn now to prebiotics, and again, I just have a selection of studies. So this is a trial of galacto-oligosaccharides at 5.5 grams per day in older people compared with placebo, intervention period 10 weeks. So this is natural killer cell activity. Natural killer cells are very important in defense against viruses and also tumor cells. And you see natural killer cell activity increased in these older people receiving galacto-oligosaccharides compared to placebo over 5 and 10 weeks of intervention. Likewise, phagocytosis of E. coli, this engulfing of bacteria, was increased significantly with galacto-oligosaccharides at 10 weeks compared to placebo. And the other thing was the galacto-oligosaccharides decreased the production of 3 inflammatory cytokines, tumor necrosis factor, interleukin-1 and interleukin-6, and they increased the production of an anti-inflammatory cytokine, interleukin-10. So as well as promoting innate immune responses, galacto-oligosaccharides seem to have an anti-inflammatory effect in this study. This is a study by the same researchers. Again, the same galacto-oligosaccharide at 5.5 grams per day versus placebo. In the second study, they confirmed an increase in natural killer cell activity, an increase in interleukin-10 production, and a decrease in pro-inflammatory interleukin-1 production, replicating the earlier findings. Again, prebiotics have been looked at in the context of seasonal flu vaccination. So this is a study we did with a mixture of inulin and short-chain fructooligosaccharides at 8 grams per day compared to placebo. These were healthy middle-aged people. They received the intervention for 8 weeks, and after 4 weeks, they got the seasonal flu vaccination. So this is the antibodies to H3N2 component of the seasonal flu vaccination. So antibodies are very low pre-vaccination. They rise dramatically post-vaccination, but they were higher in these individuals receiving prebiotics compared with individuals receiving placebo. There was no effect on H1N1, on response to H1N1 or B strain. So there's some evidence that FOS and GOS can improve immune responses and have anti-inflammatory effects in adults. Again, in the meta-analysis of pre and probiotics and seasonal flu vaccination, the authors identified 5 trials of prebiotics, and they found overall no effect on H3N2 or B strains of the vaccine, but a significant increase in titers to H1N1 across these 5 trials. So again, some evidence that prebiotics promote the immune response as manifested by response to vaccination. So these studies have used, let's call it classic prebiotics, fructooligosaccharides, galactooligosaccharides, and so on. But in the ISAP definition of prebiotics, other things were mentioned, things like human milk oligosaccharides, polyphenolics, and some fatty acids. Now, I don't want to go into the literature about HMOs, but there are quite a lot of data from both experimental systems and human studies showing that HMOs act as prebiotics. They mature the gut microbiota and mature the immune system, and that this maybe helps in defense against infection. So I think HMOs fit with this concept of prebiotics targeting the gut microbiota and having an immune effect as a result. So that's what I have here. They alter microbiota. They might have a direct effect on the immune system as well, because they might actually be able to bind to some of the immune cells in the gut wall, and they have an effect in dampening inflammation. As I mentioned, ISAT considers some polyphenolics and fatty acids to also be prebiotics. So this is just an example of one study of polyphenols. 500 milligrams of cocoflavanols per day for 4 weeks in healthy young people was shown to increase fecal lactobacilli and bifidobacteria, to decrease Clostridia, and to decrease C-reactive protein, which is sort of the prototypical biomarker of inflammation. CRP decreased from 2.7 to 1.9 milligrams per deciliter, and the change in lactobacilli was inversely correlated with the change in CRP. So these authors concluded that the effect of cocoflavanols on low-grade inflammation could have been mediated through an effect on gut microbiota, particularly the increase in lactobacilli. So polyphenols target gut microbiota. Of course, some polyphenols or a part of the polyphenols are also absorbed. They can have systemic effects, but they modify inflammation either as a result of a systemic effect or an effect of gut microbiota. Just finished talking about prebiotics to talk about omega-3 fatty acids, EPA and DHA. Of course, they have many well-described effects on human health, including reduced inflammation and in some studies improved immunity. And what's been identified in the last few years is, although I believe the main action of omega-3 fatty acids occurs systemically after they're absorbed, is that they also have an effect on gut microbiota. So they act as prebiotics. So this is one study published in Gut, 4 grams of omega-3s a day for 8 weeks, increased bifidobacteria and lactobacilli, decreased some other organisms, so showing an effect on gut microbiota. So this was a high dose, 4 grams per day. This is a low dose, 275 milligrams per day, in comparison with 20 grams of inulin per day, so a classic prebiotic. Omega-3s altered gut microbiota. They also altered serum short-chain fatty acids, including an increase in butyrate. So it seems even low-dose omega-3s may have a prebiotic action. This is the effect on different gut microbiota or different gut genuses of organisms, with the inulin group shown in green and the omega-3 group shown in orange. And it's remarkable that the inulin and omega-3s are often having very similar actions and very similar effect sizes. I think that's pretty intriguing. In the same study, the authors showed that omega-3s decrease blood pressure, blood triglycerides, and inflammatory markers, and inulin also had an effect on some of those outcomes. This is trying to show all of the outcomes and the size of the effect, with a decrease from this point here, meaning, or moving inside this point here, meaning an increase, meaning a decrease. So a decrease in blood pressure, a decrease in inflammatory markers, a decrease in blood lipids, and so on. With the fiber and in the sort of red color and omega-3s in the blue color having rather similar effects. And this is a bit complicated, but it's simply showing that the effect of the omega-3s on short-chain fatty acids, on microorganisms, was associated with the effects on blood lipids and inflammation. So they made a direct link between the prebiotic action of omega-3s and the health benefits. I'll skip that one. So there's a suggestion that omega-3s, as well as their well-described systemic effects, have an effect mediated by gut microbiota. So what about, these are nutrients, what about diet itself? So I really like this paper. It's a systematic review of dietary interventions in humans, 188 articles, putting together all of the information. So for example, here's the effects of animal protein, whey protein, and pea protein on microbial diversity, different microorganisms, and so on, and what the health effects of those different types of proteins might be. So plant and animal protein having opposing effects on gut microbiota, opposing effects on short-chain fatty acids, and maybe opposing effects on disease-related biomarkers. So in terms of diet, putting together all of the literature, looking at the effect of Western diet, Mediterranean diet, those are two I want to contrast. Different effects on bifidobacteria, with the Mediterranean diet increasing bifidobacteria, lactobacilli, different effects on other organisms, and of course different effects on disease-related biomarkers and health trajectories. So maybe some of the effect, for example, the health-promoting effect of the Mediterranean diet is due to the effect on the gut microbiota, which would be due to the nutrients and other dietary components within the Mediterranean diet. So this is a study, a figure from an ILSI Europe report looking at diet, low-grade inflammation and gut microbiota, simply gathering what I just said. So healthy eating patterns, healthy foods, nutrients that are part of healthy foods help control low-grade inflammation, have an effect on gut microbiota, and of course those two things are related to one another. So what I've told you is the immune system is the central defense system of the body. The gut-associated immune system and gut microbiota interact. The immune system certainly shapes the microbiota, but the microbiota shape the immune system. Some human trials with probiotics show enhanced immune responses. Probiotic studies show enhanced immune responses, often with lactobacilli and bifidobacteria. Studies of Goss and Foss show enhanced immune responses. I went on to talk about the fact that, let's call them non-classical dietary components, non-classical prebiotics like polyphenols and omega-3s have a prebiotic action, and this might explain some of their effects on immunity and inflammation. And diets like the Mediterranean diet that provide fiber and foods and nutrients recognized to be health supporting positively impact the gut microbiota, and this might account for some of the health benefits, particularly in terms of controlling inflammation. So there is a gut microbiota immune axis. Some probiotics enhance immune responses, prebiotics enhance immune responses, And then I talked about these other things, HMOs, polyphenols, omega-3s. Of course, improved immunity may decrease risk of infectious disease, while reduced inflammation may decrease risk of diseases with an inflammatory component. So I think gut microbiota actually has a really important public health context. So my last slide is just really discussion points. I think we need more research on this interaction. So which are the important prebiotics, including some non-classical ones? Which organisms are the important probiotics? How do effects on immunity relate to infection and so on? Do HMOs have a role in adults? Let's look more at these less traditional dietary components that have prebiotic action. Of course, people have often argued that the systemic concentration of polyphenols is not enough for them to have a health impact, but if they act as prebiotics, their systemic concentration actually isn't that relevant. I think that's an interesting point. How much of the anti-inflammatory action of omega-3s is due to a prebiotic action? We don't know. That would be interesting to dissect. And how much of the metabolic effect of food components is due to actions on gut microbiota and inflammation? So I'll finish up there, and hopefully there's a few questions that will come in. Thanks very much.